get a bad rap, but really, life wouldn't be possible without lipids. So today, let's love on lipids.
Fair warning that there's going to be a lot of jargon in this video, and I apologize, but there is a lot of jargon that I left out. Trust me. And I'll try to steer you towards what's going to be most important to focus on. Part of the reason why there's so much jargon and nomenclature is because there are a lot of different types of lipids. And so we'll talk about some of them today, but we won't even talk about all of them. And what all of these different types of lipids have in common is that they're based on hydrocarbons and they're fairly hydrophobic. Some of them are going to be more hydrophobic than others, but basically they all have this kind of hydrophobic property. And so hopefully you all will remember so well that hydrophobic effect and how, what is it driven by? Right, it's driven by the release of water, the entropy that you get when you release water that had been tied up surrounding nonpolar things. The water tries to maximize its water-water interactions. This forces the nonpolar things together, frees up water, leads to entropy. And then once you have those pushed together things, then they can kind of form those dispersion force interactions, those transient, instantaneous, just interactions between these different groups, these attractions. And so even with those hydrophobic parts, they can still kind of form interactions with one another, and we'll see how this is going to influence the properties of the lipids. How if you have really nice interactions, then what you can have is you can have that things are kind of more solid, and we can talk about fats. And then if things are more kinky and stuff, it's harder for them to interact. Maybe you have shorter chains, less hydrophobic, then you could get things like oils. And so we'll get into different properties of lipids and different types of membranes, as well as properties of membranes and all sorts of really cool stuff. I said that lipids are largely hydrophobic, and that's true, but they also, many of them are going to be amphiphilic, or what we call amphipathic. Basically, they have hydrophilic and hydrophobic parts, and depending on how they're shaped, they're kind of going to form different arrangements when you stick them in water. And we'll go into some of the different types of arrangements that these lipids can form in order to kind of have their hydrophilic parts that are exposed to the water and their hydrophobic parts just kind of clustered near one another. And so we'll talk about how we have membranes that are made up largely of like phospholipids and some glycolipids, sphingolipids, these lipids that are going to form these bilayers. So by forming these bilayers, as well as by doing other things, basically lipids have a lot of really crucial roles in biological systems. So yes, we're not really going to talk too much about detergents and soaps and stuff because we don't really have those in our bodies, but we use them all the time. We talk about them in the lab. We talk about them just like when we're washing our hands. That's another type of lipid. But we're going to focus on the biological lipids today and some of their functions. One of the main functions is compartmentalization. So actually making those membranes is really important because it allows us to kind of separate cells from one another. If you didn't have any membranes, then you would just be like a sack of liquid and you wouldn't even have the outer sac. Basically, we need membranes to form compartments inside of our body in terms of different cells, and then inside of those cells, compartments within the cell. Remember we talked about enzymes, one of the key things about them was that like entropy reduction or that proximity catalysis, how they were able to kind of like cluster things together and so that they could actually like find each other and react. Well, basically, if you had no compartments inside of your cells, everything would just be floating around, and you'd have to have like crazy concentrations of things in order for anything to happen. And then you could get side reactions and all this stuff. But if instead you have compartments and basically you can have a high concentration of some like substrate, say, inside of that compartment, but not outside of it, this is going to then allow you to kind of have specialization in different compartments and have reactions happen without having to have a ton of excess of the thing in order for the molecules to find one another. We'll also talk about how these lipid membranes, how having these compartments is going to allow us to do things like set up electrochemical gradients to drive energy making. So really, really important that we're able to have these compartments. Another key thing about lipids is they're a great source of energy storage. And so we talked about how carbohydrates, we could use like our glycogen to store energy in carbohydrate form. But as we'll discuss a little more in today, lipids are kind of even better for energy storage because they don't have to hold as much water, and they have more oxidation potential. Another thing about lipids is they can provide cushioning and insulation. So not all of the lipids have that like amphiphilic feature. So we'll talk about our simplest lipids, our triisoglycerides, which basically they're going to be like pretty much fully nonpolar, really hydrophobic. They're kind of just going to form these like lipid droplets. And then these are going to serve as like cushioning, as well as then a source for energy later. Okay, they can also play key signaling roles. We'll talk about how like the lipids, some of the lipids themselves, like PIP3 and PIP2 and stuff like this. We'll talk more about that when we get to actual biosignaling. But some of the lipids in our membranes can play crucial signaling roles, as well as some of the like steroids and stuff like lipid derivatives can play key roles as hormones. You can also have things attached to the lipids that are going to then allow for surface recognition and cells to communicate with one another, recognize one another. Really, really important functions. All of these can be played by this class of molecules we call lipids. And so different types of lipids are going to have different roles. So like I mentioned, we have those triacylglycerols, and so we'll get more into what those are, but they are mainly used for our energy storage. Then we have phospholipids and cholesterol, which is a sterol that make up our membranes. And then we have modified lipids that are used for signaling. And then within each of those, there's a lot of variability. So there's a lot of terminology, and you don't need to know all of the terminology, but there is some, and I will try to point out what is going to be important. Many lipids are derived from fatty acids, and so fatty acids are characterized by having a fatty part and an acid part. So basically that fatty part is going to be a long hydrocarbon chain. That's it. We call it its tail. These are typically 4 to 36 carbons long, and as you might imagine, that's going to be very hydrophobic. Doesn't have anything polar that water would like to hang out with. Then we have a carboxylic acid head. This head is going to basically allow for linking. So hopefully you covered in OChem how you can like join a carboxylic acid and an alcohol group to form an ester linkage. And basically this is what we're going to see is we're going to see that this head can be kind of like attached to different things. But that tail might sound really boring, but actually they can still have style. And so basically we can talk about these tails. They can have different lengths, as well as different numbers of double bonds. So if we talk about like saturated versus unsaturated, with saturated, basically it's saturated with hydrogens. So all of those carbons are bound to their maximal number of hydrogens, meaning that we have all alkane and no alkene linkages. But they can also be unsaturated. And so when they're unsaturated, basically, instead of being bound to as many hydrogens as possible, well, now they're sharing some of their electrons in a double bond. Now, this could be a cis double bond, in which case you kind of get a kink, or it can be a trans double bond, in which case you get more of a zigzag and not really changing the overall like straightness of the chain. And so we'll talk about this more. But those are different terms that we could use. Is this like saturated or unsaturated? We can also talk about the degree of saturation. So if you only have one of those double bonds, we call it monosaturated. But if you have lots, we call it polyunsaturated. And then both types can have different lengths. And so we need to have a way to kind of describe these in a more formulaic way so that we can basically know what we're talking about when we talk about different fatty acids that have different lengths and different numbers of bonds. We want to be able to kind of know what people are talking about when they're talking about a specific one. So in addition to like the super formal IUPAC notation, the systematic naming, there's also this kind of less formal notation, this delta notation or omega notation. So there's these two key types of notation. And they differ in how they're actually numbering the position of the double bonds, as well as sometimes people are kind of with the omega notation, they only tell you the position of the first double bond, because usually the alkenes are going to be separated by one methylene unit. But both of these, what they have in common is that they start out by telling you the chain length and the number of double bonds separated by a colon. So for example, if you had a saturated fatty acid that was the length of 18 carbons, you would write this as 18 colon 0. If you had a single double bond, you would write 18 colon 1. And so this would be an example of a monounsaturated fatty acid. Okay, but now where is that double bond? So in the case of this fatty acid on the right, the cis-9 octadecanoic acid, basically it's telling us that it has the double bond. This delta 9 notation is telling us that the double bond is at the 9th carbon from the carboxyl carbon. So with delta notation, you start with the carboxyl carbon and then you just count. So 1, 2, 3, 4, 5, 6, 7, 8, 9. We're at the 9th position. So we have our double bond between the 9th and the 10th. And so we would write this as delta 9. Okay, and so this is the delta notation, but what about the omega notation? With the omega notation, here we start at the methyl carbon. So here we start at the other end. And so you can remember this because omega, that's our like end, that's the end letter of the Greek alphabet. And so we start at kind of like the end of our fatty acid. And then we have to count backwards. So we would say, okay, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. In this case, it's also going to be a 9, but it won't always work out that way. When you're dealing with multiple double bonds, well, here the notation is going to differ depending on whether you're talking about the delta notation or the omega notation. If you're talking about the delta notation, well, there you have to indicate each of the double bond positions. So for example, in this fatty acid, icosapentaenoic acid, we're just going to say that EPA. In EPA, basically you have 1, 2, 3, 4, 5. You have 5 double bonds and you have 20 carbons. So you would represent this as 20 colon 5. And now we need to indicate the position of each of these double bonds. So we count from our carboxyl carbon, 1, 2, 3, 4, 5. Okay, we get a 5, 6, 7, 8, we get an 8, 10, 11, we get an 11, 12, 13, 14, 14, we get a 14, 15, 16, 17, 17, we get a 17. And then that was the end. What's kind of nice is that these are all going to be separated by 3, because basically the alkenes are typically all going to be separated by one methylene unit. So by one CH2 in between them. And so because there's kind of like this commonly, like this is what happens, like you have a double bond, then a single bond, then then a single bond, and then a double bond, and then a CH2, and then a double bond, and then a CH2, and a double bond, and a CH2. Basically, you can kind of make the assumption that these are all going to be separated out and just list the name of the first one when you're in the omega notation. But with the omega notation, remember, now you're going from the other end. So now if we wanted to name EPA in terms of omega, we could say 1, 2, 3. So this would be an omega-3 fatty acid. So there's some omega-3 fatty acids that we can't make directly, and then we can make others from those, which is why you need some of the omega-3 fatty acids in your diet. And yes. Okay, so let's practice naming some of these. So on the bottom, we see they're kind of like common names, which aren't very informative, like stearic acid, oleic acid, linoleic acid, or alpha-linolenic acid. Oy vey. Yeah, so basically, unless you memorize what each of those were, you wouldn't be able to know anything about the structure of those or the length or anything like that, unless you had some unless you have more information, such as the information that you could get if you had one of these formal nomenclature names. So let's go ahead and give them their formal names. So why don't we'll do the first one. Well, the first one's kind of easy. Okay, the first one doesn't have any double bonds. So how are we going to refer to this? We see that it has 18 carbons and no double bonds. All right, so for this, all we have to do is write 18, 0. Because it's got 18 carbons and 0 double bonds. So that's an example of a saturated fatty acid. Okay, what about the second one? So all of these are going to have 18. So we can go ahead and we can write 18 for all of these, 18, 18, but they're going to have different numbers of double bonds. So in this first example, how many double bonds do we have? We have 1 double bond. Okay, so if we were to write this in our delta notation, what we would have to do is we would have to go from this end. So we'd say 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so we have our delta 9, and we only have that one to worry about, so that's how we could write it. What about in our omega notation? Well, here too, we're still going to start with that 18,1, but now we're going to count from the other ends. So we're going to say 1, 2, 3, 4, 5, 6, 7, 8, 9. And again, we get to our 9th position. So this would be an omega 9. Okay, what about our linoleic acid? So here we have 2 double bonds. So remember that if we're using our delta notation, we need to specify the position of both of those. But if we're using omega, we just specify the first one. Right. So basically, we still have that delta 9. So now we have 18,2. We still have delta 9. But now we also have 10, 11, 12. So we have 9, 12. That would be our delta notation. In terms of our omega notation, well, here we have 1, 2, 3, 4, 5, 6. So this would be an omega 6 fatty acid. And then finally, let's take alpha linolenic acid. How many double bonds does it have? Yeah, it has 3 double bonds. So if we're using our delta notation, how would we write this? Yeah, the first one's still at position 9. So we have delta 9. And then we have that one at 12 still. But now we also have one at 15. Okay, and what about in our omega notation? Well, here we have 1, 2, 3. And now we have our omega 3 fatty acid. And so that's an example of how you would actually go about naming lipids. Okay, so like I said, things aren't as bad as they could be. So we saw that they were all kind of like separated by a single methylene group, so a single CH2. But also we typically, if you only have a monounsaturated fatty acid, that double bond is typically between that C9 and C10, so our delta 9. If we're talking about our PUFAs or polyunsaturated fatty acids, when we have multiple double bonds, those double bonds are usually delta 12 and delta 15, and usually separated by a methylene unit. So these are some examples of some naturally occurring fatty acids. So you can see in this top, so this is telling us the number of carbons and how many double bonds. We can see that the ones that I've just bracketed, these are going to be our saturated, so we have no double bonds. And then down here, we have increasing numbers of double bonds. Okay, and if we go and we look at the melting point for these, we see something really interesting. When we're just comparing the lengths, what we see is that as we increase the length of our carbon skeleton, what's happening is we're increasing the melting point. And what does it mean when we're increasing the melting point? Well, basically, that's saying it's going to take more energy to break apart the interactions between those fatty acid tails. Basically, remember how we talked about how if they could get close together, you could kind of get those dispersion forces, you could get those instantaneous dipoles, all sorts of stuff that's going to allow them to stick to one another. Once you've gotten that entropy gain from pushing them together, well, now what can happen is that the longer you have, the more area over which those dispersion forces can be acting. And so as you increase the length of the chain, you're also going to increase the ability to have those interactions. But what would happen, that was when we were just looking before, if we had the saturated. But what if we have unsaturated fatty acids? Well, especially if we have one of those cis double bonds, we're going to be introducing a kink into our chain, and this is going to be kind of disrupting those interactions. So what would we expect to see if we introduced it in terms of our melting point, if we introduced one of these cis double bonds? Yeah, we would expect that the melting point would lower. And in fact, it does. So basically, the longer the chain, the higher the melting point, but the more saturation, the lower the melting point. So when you get one of those cis double bonds, and remember that the naturally occurring double bonds are going to be in cis, you're going to get a kink. And this is going to make it harder to form those dispersion forces with one another to interact with the lipids next to you. And this is then going to make it so that you have a lower melting point. So at room temperature, saturated fatty acids have a waxy consistency, whereas unsaturated fatty acids are like oily liquids. And then the extent of how packed together these lipids can be is going to depend on that degree of saturation. So often if you have multiple saturations, multiple double bonds, that can make things even kinkier, harder to get close together. Often what we have is some sort of mixture of saturated and unsaturated fatty acids. Not all fatty acids are ideal for all purposes. And so sometimes what you might want is, say you want a more solid form of something that is an oil at room temperature. Well, you can imagine that one of the ways that you could do this would be to hydrogenate it. So basically to remove those double bonds, allow there to be more of those closer packing, allow there to be more of those weak interactions that are going to collectively make for a higher temperature, a higher melting point. So you could say, take a vegetable oil or take like a fish oil that has that omega-3 that you need, and then convert it into a more solid form. Then you can do things like make your cookies with. And so basically, scientists can do that, or labs can do that, manufacturers can do that. They can basically use hydrogen, introduce hydrogen in order to reduce those double bonds to single bonds. Another benefit of this is that basically those double bonds can undergo oxidative cleavage, which is going basically like over time, this can cause lipid-rich food to spoil when those double bonds get cleaved. So if you could basically turn them into single bonds, well, then you wouldn't have that problem. You could improve your shelf life. But what could go wrong? Well, basically, you wouldn't want to change all of those double bonds, usually to single bonds, because then you would have something that was just like a solid rock. It wouldn't be very useful for your cooking. You need something that's kind of a little more, it's kind of solidy, but more slippery. I mean, you can actually like work with it. And so typically the hydrogenation isn't done like fully, instead it's only done partially. And well, when this happens, it's going to have the effect of accidentally like having this byproduct where it's going to convert some cis double bonds to trans double bonds. So remember how I said that the cis double bonds, like all of the natural fatty acids have cis double bonds? So this is basically introducing these unnatural trans double bonds. And it turns out that these trans bonds, so these kind of the more zigzaggy ones, not the ones that actually like kink you, these basically, not only do they alter the properties of the lipids, but it also has problems health-wise. So dietary intake of these is linked to higher incidence of cardiovascular disease. They tend to raise the bad cholesterol in your blood, lower the good cholesterol, and increase the body's inflammatory response. And so this is just taken from the text slides, and I'm not an expert on that, but basically that's where this comes from. As I mentioned, lipids can be really great for energy storage. They can be stored compactly. So we saw how those lipids could kind of like get tight together. And they don't need a lot of water to do so. Basically, they're not basically because they're excluding all that water, you don't have to hydrate them. So you're not carrying around a bunch of extra water weight. So imagine if we had all of our energy stored as carbohydrates and you had to have water surrounding all of those carbohydrates. Remember how we talked about how we can make like agar and agarose gels because they soak up water real nice? Now imagine that all of the energy in your body was stored in something like that. Well, now you're basically, your body would be a big cold gel that would be all like weighted down by water. Not good. Lipids are good for this. They also give you the most bang for your buck in terms of energy storage wise. Basically, energy, we'll see when we talk about metabolism, so the making and breaking of molecules, that the way that our body does this is basically we take molecules that are really reduced and we oxidize them. So remember that when we say oxidation, that means that we're like losing electrons. And if something loses electrons, then those electrons have to go somewhere. And what happens is that as we oxidize these molecules, those electrons are getting transferred to electron transfers, like our NAD plus, our FADH. Basically, what these are going to do is they're going to take those electrons and they're going to take them to the mitochondria and use them in the electron transport chain in order to cache them in for ATP. Now, if we start with something that is really reduced, like fully reduced, like a pure hydrocarbon, that's going to be our best source of fuel because there's the most electrons that we can steal from it. So as we form those bonds to oxygen, as we basically lose electrons, what's going to happen is that we're going to use those electrons later in order to make ATP. In the case of glucose, well, here you already have a bunch of OH groups. And so you've already kind of like gone one step further than you had in the fat. And so this is why your fat is going to be able to have more, you're going to get more like calories per gram of fat than you would per gram of sugar. So we'll talk much more about how this metabolism process actually works coming up in the class. One of the inconvenient things about storing fats as energy storage as opposed to carbohydrates is that it can't be mobilized as quickly as the carbs can. So remember how we were talking kind of like about the structure of our glycogen and how it had all those branches and things where you could chew off from different points and you could quickly mobilize those individual glucose units? Not really the case for these lipids when you kind of have just these kind of like lipid droplets inside of your cells, or the plants have them like lipids inside of their seeds. Basically these big clumps of things that water's not hanging out with, they're going to be kind of hard to get to and chew up, so they can't be mobilized as quickly. But when you do get them broken up, then you do get more energy out of them. So our main form of storage lipids are going to be these triacylglycerols, or these tags. So these are going to be our TAGs. These are the simplest type of fatty acid derivative, as I mentioned. Basically, what they are is they have 3 fatty acids. So you have 3 tails. And then they're linked through esters to a glycerol backbone. And so basically, this is going to be a glycerol. You do need to know the structure of glycerol. Super simple. You've just got 3 carbons. And then they're attached to, basically, each of them is going to be attached to an OH. So you have a triol. And basically what's going to happen is that, going back to our idea that we can kind of have a carboxylic acid group interact with an alcohol group to form an ester. So our ester, this is going to be what we're going to see here. So we have these ester linkages. So remember that an ester is just a carbonyl carbon next to an oxygen next to another carbon. And we call this a triacylglycerol because the acyl, basically that's referring to the part of, it's referring to basically when you have a carbonyl carbon attached to something, we call that an acyl. So this is like an acyl, triacylglycerol. So you have these 3 acyls that are going to be attached to your glycerol. Now, these 3 tails, they can be the same or they can be different. So if we look at this example over here, basically we can see that we have 2 of them are going to be saturated and 1 of them is going to be unsaturated. And in this case, what we would call it was, we would say it was mixed. So if it was simple, these would all be the same, but this is mixed. So we have 3 different fatty acids. And so they could have different lengths too, and different numbers of double bonds. What these triacylglycerols have in common is that they're very hydrophobic. So basically you just have pure hydrocarbons. Even in the backbone, because you're using up those OHs to form these ester linkages, this is going to be very, very hydrophobic. And so these are going to be very water insoluble, which is how you can get those things like those lipid droplets. In terms of natural fats and oils, these are typically a mixture of simple and mixed, saturated and unsaturated tags. So if you're thinking of like olive oil, which is going to be a liquid at room temperature, well, it's predominantly going to have the C16 and C18 unsaturated. And so when we say unsaturated, remember that's telling us it has those double bonds, those kinks that are going to make it more fluid, so it makes sense that it's a liquid at room temperature. It has those double bonds that are reducing the melting temperature. When we talk about things like beef fats, that's going to be hard and solid. Well, now you see you have a lot of these saturated, tags, which are going to be able to form those stronger interactions with one another, harder to break apart. You need more energy in order to do so. So you have a higher melting temperature. At room temperature, you'll find them being this like hard solid. In terms of a butter, well, now you have more of a mixture. And so you can see that basically the different properties of fats and oils are characterized by the different properties of the triacylglycerides that make them up. Now let's talk about some more fun lipids. So yeah, those triacylglycerols are cool that they were doing all that energy storage and making cushioning and all that nice stuff, but they were kind of just like sitting there as blobs before then. But what about our membrane lipids? Well, basically, the reason why those triacylglycerols were kind of sitting around as blobs is because they're just like really unreactive. They're basically mostly just hydrocarbons. They are really not non-polar, just like fully almost. But what if we have something that is partly hydrophilic and partly hydrophobic? Well, in this case, instead of just forming like a clump of nonpolar hydrophobic stuff, now if we were to stick those into water, we can do things like form membranes. And so we can have, say, a phospholipid, which has a hydrophilic head and hydrophobic tails. We can put that into water and get a membrane. And so let's talk about our membrane lipids. So when we talk about membrane lipids, so remember, what do they have to have? They've got to have that hydrophilic head and those hydrophobic tails. And so when we have something like this with hydrophilic and hydrophobic parts, we call it amphiphilic. So amphi for both, and then philic for loving. We can also call these amphipathic. And the key ones that we're going to talk about are phospholipids, which have two fatty acids joined to glycerol or sphingosine. So in the case of our triacylglycerides, we had 3 fatty acids joined, 3 of those tails. And in the case of our phospholipids, we're just going to have 2 tails. But then basically one of the other carbon in the backbone is going to be used to form a bond to something that's polar. So it can be polar, it can be charged. And so that's why we kind of have 2 tails, because well, one of the glycerols is being used, one of the alcohol groups from the glycerol is being used to make this connection to our hydrophilic head. Additionally, we can have phospholipids that instead of having a glycerol backbone, they have this thing called sphingosine, and so we'll talk about it in a minute. Now, these phospholipids are going to be interspersed with other things like glycolipids, where basically they have a simple sugar or a complex oligosaccharide. So one of those like glycan things at their polar ends. So basically it's just sugar on a lipid. So you can get sugar sticking out of your membrane, like things like those ABO blood types, sugars that determine that. It's like we talked about. Now they're also sterols. So our key one is cholesterol. And these have a rigid system of 4 fused hydrocarbon rings. And then we'll go into how the structure of that cholesterol is going to help keep our membranes fluid. So basically, this is a little intimidating of a chart. So hopefully I can kind of steer you towards what we're going to focus on. So let's start with those phosphoglycerides, our glycerophospholipids. And remember that those are going to have our glycerol backbone and attached to 2 fatty acids. Through 2 of those positions, 2 of those OHs, and then the third position is going to have a head group that is going to be linked through a phosphate. And so the 3 key ones that you need to know, you don't need to memorize their structure, but you should know the names. ethanolamine, choline, and serine, in which case we would call it phosphatidylethanolamine, phosphatidylcholine, or phosphatidylserine. As for those tail positions, well, just as we saw before, these can kind of have like different lengths and different degrees of saturation. Thankfully for us, things are kind of simpler. Instead of all those different combinations, you typically have in the first position, like a C16 or a C18 saturated fatty acid. And then at that C2 position, so that second position in our glycerol, you typically have a C18 or C20 unsaturated fatty acid. So remember, unsaturated means it has those double bonds, it has those kinks that are going to make it so that the chains can't get as close together. And this is going to make it so that our membranes have some fluidity, since we kind of have one of those straight chains and one of those kinky ones. And then in the third position, that's where we're going to have our highly polar or charged head. That's going to be attached to the third OH position in our glycerol through this like phosphodiester linkage. Those head group constituents, remember, that's the part that can vary. And so we have things like our phosphatidylethanolamine, phosphatidylcholine, and phosphatidylserine. But then you can get all sorts of other derivatives. In terms of our other type of phospholipid, our sphingolipids, basically here for their backbone, it's also one of their tails. So it's a little confusing, but sphingolipid, instead of having a glycerol backbone and two tails, two fatty acid tails, it has a single fatty acid tail, but it also has a tail that's coming from the sphingosine. So the sphingosine itself, it kind of has this L-shaped. And so it takes the position of that glycerol, where you have that one position is attached to your head group. Then you have one position that's going to be attached to a fatty acid. And then the other position, it's kind of just attached to the hydrophobic chain of sphingosine. So this molecule sphingosine is what our backbone / tail is for one of them. And then we have, instead of having an ester linkage, we have an amide linkage, because we have it attached through a nitrogen. There are different types of things that then can serve as our head groups. Just as we saw before, we could get things like a phosphoethanolamine group or a phosphocholine group. But we could also get things like sugars and like simple sugars, single sugars, monosaccharides, which we have like a cerebroside. We could have things that are really complex, which we call like gangliosides. So there's all these different types of names and all these types of sphingolipids, and they play really important biological roles. I don't need you to know all these names. They have important roles in your body that we're not going to be able to get into. But as the myelin might suggest, these are going to be really important for like myelination and things in your neural tissue. They're also important for cell signaling and cell surface recognition. So we talked about how some of the sphingolipids had those like sugars attached to them. And before we had talked about how like some of the sugars attached to your cells are going to allow your cells to kind of like recognize one another and to see like that's recognize other things as foreign, such as other people's blood. And that was because they had those different carbohydrates groups displayed on the cells. And one of the main ways that they're displayed on the cells is by attaching them to these sphingolipids. And so they play these key biological roles. Again, we just don't have time to get into them. Okay, we do need to, though, get into our sterols. So remember, our main sterol we're going to be talking about is our cholesterol. Well, what all sterols have in common is they kind of have this core structure of 4 fused carbon rings. So this like A, B, C, D. This is showing you the structure of cholesterol, but there could be other sterols that still have this structure. In addition to this like core, which is almost planar and fairly rigid, it also has this like alkyl side chain, as well as a polar head group that's going to allow it to kind of have a little polar part that can stick out into and hang out with the hydrophilic heads of those other guys in the water, but then kind of like hide the rest of it in with all the nonpolar tails of those others. Additionally, sterols can form as precursors for hormones, testosterone, cortisol, prednisone, aldosterone. All of these are basically steroids, which are just oxidized derivatives of sterols. So you can see that they still have those like 4-ring structures, but these are going to have more oxidation to them, and they're not typically going to have like that long nonpolar tail. And these can form important things like hormones, various cool stuff. Speaking of various cool stuff, there's a lot of types of lipids that we didn't get into and that we're not going to get into, but your book goes into if you're interested. You can things like eicosanoids, which are going to serve as paracrine hormones, such as prostaglandins and that sort of thing. We'll talk a little bit about some of the intracellular signaling roles of some of the lipids like phosphatidylinositol and sphingosine derivatives, things like our PIP2 and PIP3. We'll get into some of that. But for now, you don't need to worry about that. But know that there's a lot of cool stuff about lipids. I'm also not going to talk about waxes, but basically waxes are kind of just esters of long chain fatty acids that allow you to have things like bunny bees do cool stuff. And similarly to other lipids, they're going to be that hydrophilic, they're going to repel water. In the case of a wax, it's kind of going to have this, well, waxy consistency. So they have, as Anna mentioned, they're going to have like a higher melting point to keep them that solid, that really hard solid. Okay, but let's talk about things that are less solid. Like we wouldn't want our membranes to be solid. Instead, we want our membranes to be fluid and we want our membranes to be dynamic. And we need to talk about what's going to happen when our amphiphilic lipids meet water. Not just that super hydrophobic wax that's just going to repel the water, but our amphiphilic lipid, which has that hydrophilic part, which will hang out with the water, and that hydrophobic part, which will be excluded by the water. What would happen if you put that into water? Well, it depends on the shape of that amphiphilic lipid. If you have something like a soap or something like a detergent, where basically what you have is you just have a single tail and a head, where the cross section of that head is going to be greater than that of the side chain. You kind of have like a coney thing. If you stick that into water, what's going to happen is it's going to form this thing called a micelle, where basically all those nonpolar parts are kind of just going to be in the center and there's no room for anything else around it. So there's no liquid inside of here. We have the single layer and it's just like this blob of gunk inside and then this hydrophilic outside. So kind of like one of those, what is it? Like jawbreakers or what are those like those candies that have those like hard outer but then soft like inner? I don't know. You know, hopefully what I'm talking about. That would be like your micelle. And we saw how we could actually take advantage of micelles in the lab to do things like purify out membrane proteins because they form that nice kind of little lipidy bubble thing. But that wouldn't be very good for our cells if we couldn't have anything liquidy inside of them. And so instead, we need something that instead of having that shape like a cone, we have a shape that's more like a cylinder. And where did we see a shape that was more like a cylinder? Well, with our phospholipids, because remember, we've got those two tails there instead of just a single tail. So we have this more cylindrical shape. And now it's not going to be able to form that micelle because it can't kind of compact that way. Instead, if you stick it into water, what's going to happen is it's going to form a bilayer. So it's going to form this kind of like sandwich where those hydrophobic tails are going to be in the center and the hydrophilic heads are going to be on the outside. And if you have enough of this that it like folds up on itself, well, now you can get some sort of like vesicle where you actually have liquid inside. And then you have this bilayer membrane. And you can imagine that, so this would be also called like, these vesicles are sometimes called like liposomes. You have these like small ones, but now imagine that you have a lot of liquid in there. You can get these really, really big compartments. You can have cells. And then inside of cells, you have all sorts of different compartments. So inside of our cells, there's actually like a huge interconnected network of membrane-bound compartments. So we have those like bigger things like our mitochondria, our nucleus, our endoplasmic reticulum, and our Golgi apparatus, which is where the kind of like some proteins are processed and stuff to be made, to be secreted, or otherwise modified. But then we have smaller compartments like our lysosomes, which break things down, and endosomes, secretory vesicles. We have all sorts of these membrane-bound things that are kind of all interconnected through this dynamic endomembrane system. And different membranes within that system actually have different compositions. So we saw all that variability we could have with our fatty acids and with our phospholipids, with our glycolipids, all those various things. And they're going to differ between different compartments in the cell, between different membrane-bound compartments, and even between the two leaflets of the bilayer. So when we talk about that bilayer, we talk about like the outer leaflet, the one that's actually facing the exterior, and then the inner leaflet, the one that's facing the interior. We call those leaflets, and those can have different compositions of lipids. And so you can see that, say, like in your plasma, the plasma membrane, so the outer membrane, we have a lot of cholesterol, less of our like phosphatidylcholine. But then if we look at something like our nucleus, we've got a ton of phosphatidylcholine and not as much cholesterol. So how do we balance this stuff all? How did this get made? How do these get made differently? We need some way that these lipids can actually move around. So basically, lipids can kind of diffuse laterally without help. And this can happen really fast, so they can move like 1 micrometer per second. And of course, this is all just kind of like assuming that there's nothing in the way. But of course, as we'll look at later, there's going to be a lot of stuff in the way, a lot of proteins and things like this. So they couldn't just like diffuse totally freely. But if they could, they can move very fast in this lateral direction within the leaflet. But it's actually really hard to go a flip-flop. Because remember that it's going to have that hydrophilic head and that hydrophobic tail. In order to kind of like flip through, it's got to get that hydrophilic head all the way through all of this nonpolar stuff onto the lower lip. So it needs help. And so there are proteins in the membrane that are going to help it out. And they have really cool names. You don't have to remember which is which, but they are really cool names. So a flipase is basically going to move your phospholipid from the outer leaflet, so that one that's facing the outside, to the inner leaflet. And then you have a flopase, which kind of goes the opposite direction. And so it's moving those phospholipids from the cytosolic leaflets to the outer leaflets. And then you have a scramblase, which is going to basically move things in either direction towards an equilibrium. And so these are like calcium dependent and things like this. So that would be your scramblase. But we have your flipase is going to take things out or in, and your flopase, which is going to take things in or out. And by doing this, you can help kind of specialize the different membranes so they can better fit the functions and needs of the different compartments inside of the cell. And so that you could basically do things like have signaling, you could do things like have different membrane proteins displayed, different sugars displayed, and things like this. Various cool stuff that can come because you're able to do this flipping and flopping and scrambling. But just like we saw that those proteins were doing that flipping and flopping and scrambling, there's a lot of other proteins. Cell membranes are not just lipids. And in fact, about half the mass of some of our membranes comes from proteins. So the lipids are smaller though. So there's about 50 times or more. There are about 50 times or so more lipid molecules than protein ones. But in terms of mass, about half of that mass is going to be coming from proteins. So you're going to have big proteins interrupting all those lipids. And in fact, there's this thing called the fluid mosaic model that basically describes a pattern that's formed by individual lipid and protein units in a membrane. And that basically this pattern can change while maintaining the overall properties. So inside of this membrane, we have all sorts of stuff. We have proteins attached, proteins going through, proteins attached to either side. We have those sugar chains attached. We have all sorts of stuff going on. And basically, though, this is all very dynamic, is kind of what the point of the fluid mosaic model is saying. It's saying that there's a lot more than just lipids in the membrane, and that what's happening in the membrane is very fluid and it's dynamic and it's changing all the time. And it turns out that even this fluid mosaic model is too simplistic. Instead of being able to like freely diffuse around, what happens is that actually there are various like regions or microdomains of the lipid membrane. So about like 50% of it is like what we call these lipid rafts or microdomains. These are basically clusters of where the lipids are enriched in cholesterol and sphingolipids. This part is going to be like slightly thicker and more ordered than the neighboring region. So remember that cholesterol's got that kind of like stiffer structure. It's going to hold things kind of like stiffer. And so this region is going to be like more ordered. It'll also be enriched in various types of proteins. Speaking of which, those proteins can't just be freely diffusing all over the place. either, because basically, in addition to some of them being anchored to the outside, a lot of them are anchored on the inside. And so this can do things like help with the curvature of membranes and things like this. It's also going to make it so that these membranes that these proteins are kind of going to be restricted in their movement. So some of them can move more than others, but some of them are really fairly restricted in the membrane. But you can imagine that this can be kind of helpful to have various kind of compartments, or not compartments, but various regions where things are concentrated at various parts of a membrane. It also makes it so that your cells can regulate what is on the membrane more easily. If you can say have these things concentrated one place, that then you can kind of like pinch in that part of the membrane. And you can pinch in that part of the membrane because you have things like those various proteins helping anchoring things inside to kind of pull things in. And so if you want to know more about all that, there's a lot more in the book that I just don't have time to get into. But membranes are a lot more complicated than we've given them credit for. And more research is showing that. But for now, let's go back to the simplistic view. Yeah, I know it's not simplistic. There's a lot of terms, and I apologize. But going back to our lipid part, basically the fluidity of that membrane, the ability for things to actually have that fluid mosaic model is going to be dependent on the membrane, not just kind of being stuck in place. And so if we had membranes that were made up out of that like triacylglyceride, well, not only would it not have that hydrophilic head to do things, but if you had those three tails that were coming really close together, if you had a fully saturated membrane, what's going to happen is that you're going to have that tight packing. You're not going to be able to do that diffusing. You're not going to be able to move things around easily. You're just going to have like a solid clump, which would not be good. And so we want something that's going to be more fluid. And so that's why we have things like we have our unsaturated fatty acids in our membranes. We introduce those kinks. So remember, you typically have one saturated and one unsaturated fatty acid per phospholipid, so that we can get those kinks, we can get some of that fluidity. And we also have that sterol. We're going to have that cholesterol that we'll talk more about in a second, because it's going to play a key role in dictating the fluidity. What also dictates the fluidity, though, is temperature. So even if you have that beef fat, if you get it hot enough, you can melt it. Similarly, instead of talking about like a melting temperature, when we talk about our membranes, which are this like heterogeneous mixture, so basically you have a bunch of different stuff in there, we talk about this like transition temperature, where basically below that transition temperature, things are kind of more like solidy, they're kind of like stuck in place. But then above that, you get this more disordered. You get things moving around, because remember, what is temperature? Temperature is just like heat, it's energy. It's the amount of like energy that they have. And so if they have more energy, they're able to move around more, they're going to have more fluid composition, more disorder. And so that's going to, if you have an energy that's enough for that, that's what you're going to see. But below that, you're going to see kings kind of just like stuck in place more. And that transition point, we call the transition temperature. Now we call it a temperature, but really it's not like everything goes from one thing to the one to the other like instantaneously. So instead it's more of a kind of like range. But what happens is if you just had phospholipids, you would see that you would have this kind of sharp inflection point at your TM. And basically what this would mean was that you didn't have much of that wiggle room. And so if you were to go slightly above the TM, well, now you would have everything kind of be all liquidy, all like oily kind of. But if you were to go below it, you would have things would be all solid really quickly. And so you wouldn't have much of a buffering area. You could quickly get yourself in trouble, have your cells all melty, or have your cells all like stuck in place. And so we need to add this sort of buffer. And the buffer that we're going to use is going to be our cholesterol. So not a pH buffer, we're not talking about pH here, but just a buffer in terms of kind of allowing ourselves a little bit of safety space so that we don't have that sharp transition. In this case, what we're going to use is our cholesterol. So remember, cholesterol was our sterol. An example of a sterol, it's got those 4 rings, and it's got the hydrocarbon tail, and then it had that OH on the end. It got that slightly with that one little polar part, so that it could hang out with those polar head groups. But then what's going to happen is that non-polar part is going to be interacting with the phospholipid tail next to it. And because this ring system is going to be less flexible, it's kind of going to stiffen up this region. Then you're still going to, at the end, kind of get a more fluid region. But you can think of this cholesterol is kind of like interacting with these things. And so what's going to happen is that basically, if you're at high temperatures, when all the stuff around it is able to be really flexible, when all of this region of the phospholipids kind of like moving all around, well, here the cholesterol, its ring system can't move around easily. And so this is going to help stiffen things up when you're at those high temperatures. And it's going to reduce the fluidity at those high temperatures. It's going to make it so that you have to have a higher TM in order to get that same amount of fluidity. But what happens at low temperatures? I mean, like in order for a buffer to be a buffer, we need it to be helpful at low temperatures too. And at low temperatures, we would need it to increase the fluidity. And it does this because what it's going to be doing is it's basically interrupting the packing of these phospholipids next to one another. So at lower temperatures, it's kind of preventing things from forming those interactions that would hold things in place. And instead, it's kind of going to be making it so you have a lower melting temperature. It takes less energy to make things fluid. But then when things are starting to get all fluid, well, now that cholesterol isn't getting all fluid. And so now it's going to be harder to get it even more fluid. It's going to be harder to kind of make it oily. And so this cholesterol is going to act as this important fluidity barrier. So I know that was a lot of terminology and a lot of concepts. So remember that you're not responsible for all of those different names, but you are responsible for knowing the key roles that lipids can play. And unfortunately, to know some of those key roles, we kind of have to know some of the names and some of the nomenclature because we have different types of lipids playing different roles. So we have our triacylglycerols. Those are going to be used for our storage. Those have those glycerol backbone and those 3 fatty acid tails. Then we had our phospholipids and cholesterol that were making up the membranes. We had the phospholipids and we had our glycerophospholipids and our sphingosphospholipids with the glycerol or the sphingosine backbone, and then those 2 tails, and the hydrophilic head. And then we had all sorts of modified lipids used for signaling and a lot of variability within each type. But all of these are going to allow for things like compartmentalization, having those phospholipids and glycolipids that are making up our membranes with the help of sterol, kind of keeping things fluid, providing those triacylglycerols, forming those energy storage and cushioning and insulation. And then we had our signaling as well as organization, having sugar chains and having proteins attached to the membrane proteins. So all sorts of really, really cool stuff about lipids, all sorts of stuff we didn't even get to, but I think that was enough for today. So hope that helped.